like the promoter or the control elements, it probably would end up decreasing expression. If you wipe out the TATA box and the CAT box or the control elements, then those transcription factors and activator proteins won't have anything to bind to, and so you'll probably end up decreasing expression of the gene. But again, by random changes, you could in some way improve those sequences, create sequences that the transcription factors bind to better, so you might actually end up increasing the expression of the gene. So anyway, I guess the point here is that on the surface of it, it seems like these things are bad things because they could potentially be gene wreckers. And there are, when they analyze our genomes, they do find some evidence of genes that were wrecked by these transposable elements. Remember that a lot of recessive diseases are caused by non-functioning alleles of a certain gene. And there's a genetic disease called hemophilia, where people bleed for a long time because they don't have a functional copy of one of the clotting factors necessary for blood clotting. And at least for some forms of hemophilia, when they look at the recessive allele, the alleles that the person has, they can see that what caused the allele to become recessive was a transposable element that just landed right in the middle of the clotting factor gene and blew it apart like you see there. So anyway, there is some direct evidence that they can wreck genes. But at the same time, they can also end up duplicating our genes. These transposable elements can end up being like photocopy machines that make extra copies of our genes. And here's the mechanism for that. Imagine that there's some good and functional gene that's sandwiched between two transposable elements, like you see here. Well, when the transposable element on the left gets expressed, when a messenger RNA gets made from the one on the left there, oftentimes what can happen is the RNA polymerase II doesn't necessarily stop there, and will keep rolling along and make a gigantic messenger RNA that includes not just the original transposable element, but the gene between that and the second transposable element. So you get this big messenger RNA like that. And then when the reverse transcriptase, here we go, comes along, you're now going to get a copy of the original, or that gene that was sandwiched between. And so when this reinserts itself, like up here, like there, notice you end up with a new copy of the gene like that. So, we normally have 2 of every gene because we're diploid creatures, but if this happened to one of your genes, you'd now have 3 copies of that particular gene in your genome. So does having an extra copy of one of your genes do you any good? Well, it might, and I'll tell you how it might in a moment, but before I go on with that, there's a second way that you can end up with extra copies of genes in your genome. And it's something called unequal crossing over. We talked about crossing over when we talked about meiosis. That is, before one of your cells makes egg or sperm, then in other words, before it goes through meiosis, each of the homologous pairs of chromosomes, like your 2 chromosome number 1s, for instance, line up with each other and do something called crossing over, where segments of each homologous chromosome can exchange places with the other homologous chromosome, like this. And normally when crossing over occurs, it's called equal crossing over. In other words, the size of the piece that breaks off on one is exactly the same piece that it exchanges with the other. So each chromosome does not gain or lose any genes through normal crossing over. But if it's this unequal crossing over, as sometimes does occur, it looks like this. The pieces that exchange with each other are not equal size pieces. And so notice that the chromosome on the bottom has actually ended up gaining a new copy of one of the genes, gene B in this illustration. And so the offspring that inherits this chromosome on the bottom right there is going to end up with 3 copies of gene B, 2 from this parent and 1 from the other parent who had a regular chromosome number 1 with just 1 copy of gene B. So anyway, the point here is that transposable elements are one way we can get duplicates of our genes, but unequal crossing over is another way. And they're not actually 100% unrelated, because what they found is the more transposable elements you have in your genome, the more likely you are to get unequal crossing over. Somehow having the same sequence, the same transposable element sequence in your genome somehow fools the chromosomes, and they're more likely to do unequal crossing over. So the point here is that transposable elements can, through various ways, lead to extra copies of genes in the genome. And that leads to an interesting phenomenon. Once a person has some extra copies of genes in their genome, either through errors in crossing over or transposon moving them around, the extra copy can then undergo mutations. Now, all genes can undergo mutations. Mutations happen randomly just because there's radiation in the environment we're constantly getting hit by during the chromosomal duplication processes. Sometimes DNA polymerase III enzyme makes a mistake and introduces mutations into the DNA. There are mutagens in the foods we eat. Even if you eat healthy foods, there's always going to be some chemicals that can cause mutations in there. So all our genes are constantly being subject to random mutations. But the thing here is, if you have an extra copy of a gene, it doesn't hurt you if that gene mutates, because you're not really using the extra copy of a gene. It's an extra copy. To make an analogy, if you have your car key and you make an extra copy of it, you can mutate that new car key any way you want to, and it won't stop the ability to use the original car key to start your car and lock up your car. So in a way, this gives us having an extra copy of genes is good for us, because it makes a new gene that is free to mutate, and so we might, through that new gene, be able to get some sort of, if it mutates in a good way, get a new protein out of it. Now, remember, most mutations are random, so most of the time you'll end up inactivating that new copy of the gene, and that's sometimes called a pseudogene, one that's very close in sequence to a functional copy of the gene, but one that is different enough so that it's not expressed. But as I said, you occasionally you could get a newly functional gene. Now, if you notice on the lower chromosome here, when this happens, the 2 copies of the gene are right next to each other on the chromosome. You notice the 2 gene Bs right next to each other. It doesn't necessarily have to stop with just 2 copies of the gene. This process could continue. You might have 4 or 5 copies of the same gene all in a row. And they find those actually throughout our genomes. They're called multi-gene families, a group of identical or at least very similar genes at one location in the genome. And it's not all that rare. They think about half of the genes that we have are part of multi-gene families. And a really good example of that are the hemoglobin genes. I've been talking about hemoglobin throughout most of the semester. It's found inside our red blood cells. It's the protein that holds oxygen. The little blue dots there are supposed to be the hemoglobin proteins. The actual hemoglobin protein, which is depicted here, is one of those proteins that's made out of more than one polypeptide chain. There's 2 types of polypeptide chains. One's called alpha globin and one's called beta globin. And they come together to make the fully functional hemoglobin protein. But when they look at the human genome, they find that the alpha globin gene is part of a multi-gene family, and so is the beta globin gene. Here's the beta globin gene family. I guess it's on chromosome number 1, and the dark blue bands there are functional variations of the beta globin gene. As you can see, they're expressed at different times in our development. And the green colored one there is a pseudogene. It's very similar in sequence to the other ones, but it's been mutated to such an extent that it's just not expressed. And the alpha globin gene also comes from a multi-gene family. Here again, you see the blue ones are expressed forms, functional genes, whereas the green ones are pseudogenes. I guess this one is on chromosome 16. And just kind of a tangent thought just reminded me looking at this. Remember that in bacteria, proteins that function together are often grouped in these things called operons, where all the genes for the proteins are clustered together. Whereas I told you in eukaryotes, even genes that function together, the genes for proteins that function together are not usually clustered together. And this was a good example. Alpha globin and beta globin function together to make hemoglobin, but the beta globin part is on chromosome number 1, and the alpha globin gene comes from chromosome 16. We just don't have operons in eukaryotes. Anyway, so the point I'm making here is that they think these multi-gene families are related to the high number of transposons that we have, because transposons directly or indirectly lead to copying of genes. And so does it do us any good to have a bunch of these extra copies of the genes? And I guess the answer to that would be yes, because having all these different copies of the alpha globin and the beta globin genes has allowed these genes to mutate into specialized forms for us. As you can see from the diagram, certain types of these hemoglobin genes are expressed at different developmental stages, some in the embryonic stage, some in the fetal stage, and some in the adult stage. And that does have a beneficial effect for us. When the baby is developing inside the mom, the baby needs oxygen also, obviously. And the embryonic and the fetal forms of hemoglobin have a higher affinity for oxygen than the adult hemoglobin in the mom cells. And that's good, because then it allows the baby to extract more oxygen from the mother's blood than would otherwise be possible if they were using the same form of hemoglobin. I guess the point here is that having a bunch of extra copies of a gene is a good thing, because it allows you to evolve new proteins with new variations in the function, like we see for the hemoglobin here. And what brings us these extra copies of the gene are these transposons that we have, because they're related to making extra copies of the gene. So I guess I'd say that all these transposons in our genome, we have a huge amount, 44%. They're harmful and they're helpful. They're harmful in that they can wreck genes when they insert in them, but they're helpful because they can be gene copy machines, and that perhaps has allowed us to evolve more proteins than other species that have fewer transposable elements. So maybe we are a very advanced species, evolutionary-wise, because we're burdened with this huge amount of transposable elements in our genome. Sort of an interesting concept to think about. Well, any questions on the transposable elements? I'm running out of battery power. That's perfect timing, I think. All right, well, let's see. Well, that about wraps it up for this lecture on genomes. Oh, actually, I take it back. There was one more bit I was going to talk about, but I kind of skipped over. Up to this point, I've been emphasizing how the transposable elements can end up being copy machines to give us extra copies of genes. The transposable elements also can do the same trick, but with exons, individual exons instead of entire genes. So imagine, as you see in this diagram, that there's an exon that ends up getting flanked by two transposable elements. Well, when the transposable elements make their mRNAs of themselves, they're going to end up making including an mRNA one for the exon, and when the reverse transcriptase comes along, you're now going to get a new extra copy of that exon in the DNA, like you see there. And so when this thing inserts itself, it could potentially bring that new exon and insert that new exon into a already existing gene. And so it can do what's sometimes called exon shuffling. Let's see something here. Gene cloning there. Huh. Well, now I don't see it. But anyway, no, well, I lost the overhead. But anyway, so this is called exon shuffling. And it can help piece together new proteins, because remember that individual exons tend to be functional domains of protein. Like an individual exon might be a hydrophobic transmembrane domain, or it might be an ATP binding domain, or it could be a domain that sticks out of the cell membrane to bind glucose or something like that. So by bringing in new exons, you might be adding new functional elements to proteins that you already have. And there is some evidence of this. There's a gene in our genome called the tissue plasminogen activator gene, or TPA gene. It's the one you see there on the right. And just looking at the diagram, it looks like this gene has 4 exons. The one they call F, E, G, F, and the 2 ones are K there. And when they look at other places in the genome,